Okay, so let me go over through the sequence of events for today. This is the last day of our second unit, our genetic processes unit. I'm going to start off by taking up the homework questions from the blood typing activity from yesterday. I'm hoping people got to that. I realized I was pushing a little bit how much we can actually accomplish in a day. So in order to like stay on timeline, we had to do a little bit of extra. But I'm gonna, like I said, I'm gonna start by taking up the homework questions from that. And if people have any other questions about the multiple allelic inheritance, ask away. Okay, so we'll take a moment to do that together. The second part of this morning, you're going to be doing a little bit of reading on genetic illnesses. So it's page 204 to 207 in your textbook. And it goes along with this sheet in your course notes. There's a little section in your course notes on page 14 called genetic disorders. Okay, so you read this little blurb on genetic disorders. And then there are some questions from the textbook that you're going to answer on genetic disorders. It's these guys right here. It's on page 207, 1, 2, 3, 4, and 5. And depending on how fast you read, this is not a super long activity. So I've done that intentionally. We'll keep this fairly brief because I want you to have as much time as possible to work on the project. So what is the project, you may ask? Well, there are actually a few things that are coming due. So the first thing that you need to finish by tomorrow, ideally, would be your vocabulary list for this unit. Okay, so you should be well on your way. Hopefully you're adding words as you go every day. That certainly makes it way easier if you're doing that during learning block 3. But you should be on the way to finishing that genetic processes vocabulary list. So ideally I'd like to get that from you today. But today, tomorrow, this is fine. I'm probably not gonna get to marking these until the weekend, so not a huge deal if I don't have it until Friday, but again, you probably don't want it to bleed over into your other class. Yes?
When are we getting our marks back for the slideshow?
I'll get back to you. I'm drowning in stuff right now. I've got so much stuff. By the time I finish preparing the lesson for the next day and move everything over to this thing and make new things and stuff, I'm way behind. I'll get back to you on that. I don't have a good timeline for you. Soon, hopefully. The other thing today is the quiz. So you mentioned this already. You can find it right there. So I think it might be invisible to you actually. It becomes visible at 12:50 automatically. So you'll see it there at the beginning of learning block 3. So the quiz is just like the first quiz in that it's a mix of multiple choice, choose, check all that apply, okay? It's a little bit longer than the first one, so I think there's 38 questions here. It's still open book, okay? So you can use your notes, you can use whatever resources you want. I did put a time limit on this one of 2 hours, which is not a really strict time limit, okay? That's a very lackadaisical time limit. But the reason I put it on there was I looked over some of the last quizzes and some people took 6 hours to do the last quiz. That's not really the intention there, which is that you're just kind of doing it in the background and taking a really long time to look up an answer to every single question. That probably means that you don't know the answer to any of those questions, which is not the idea of the quiz. So I don't want it to be infinite time where you just you look up every single thing and take 6 hours. That's excessive and probably makes the test or the quiz meaningless. So I did put a 2-hour time cap on there. If you have extra time, if that's part of your accommodations, that's automatically built in for you so that it'll just give you extra time. But please be aware there is a 2-hour time limit and we're approaching the 2 hours. I think it's 5 minutes away. It does give you a little beep and say oh, in 5 minutes, you gotta hand this in, don't forget to hand it in, okay? Please, abide by the 2-hour time limit, okay? I mean, obviously, it still lets you submit it, but it only checks off the stuff that you did before the 2 hours. That's how it works. So if anything you add after the 2 hours, it still puts it on there for you, but it just doesn't count it. So it'll give it to you as a mark, but when I get the mark, it doesn't include those ones that you did after the 2 hours, just so you know. So don't go over the 2-hour time limit. It should be plenty. I mean, it's several minutes per question. If you need more than several minutes per question, you definitely don't know the answer to that question, okay? So Just as a heads up. I did reduce it a little bit. We'll see how it goes. Okay, if it's really onerous for people and people are like, ugh, like I can't do this in 2 hours, then let's reevaluate. But I think it should be very reasonable. So, okay, so then that's the quiz. That like I said, that's for learning block 3 today. What people are probably curious about is what the heck is this assignment? So let's talk about the assignment in more detail. Why can't I see the details of this? How do I do that? What I want to do is click up here. Why can't I click up there? Is there any way to view this full screen on an iPad? Nobody knows the answer to that. I'm going to figure this out. Okay, switch gears. What if I do this? Does that work? Yes. Cool. Okay, so people at home can see this. Good. This will work. All right. Now I can view it as a student. I don't know why it does that. It doesn't let you look at the assignment. It only lets you like see that you made one when you click on assignments as a teacher, which is really weird. How is this live? Here we go. Okay. So the purpose of this, I mentioned this yesterday, is to create a learning tool to teach somebody else the process of meiosis. And the idea here is that you're going to show me the process happening. Okay? So ideally, you want to be able to have some kind of a cell on screen that you can manipulate the parts of, drag stuff around, so you can show me what is actually physically happening in the cell. Okay, that's the idea. Now, I've realized that the Chromebook is like not the most, what's the word I'm looking for here? Robust system for like running such programs, okay? And many people only have access to a Chromebook, which I totally get. So we're gonna be a little bit flexible here in terms of like what this is going to look like. What it's gonna look like is kind of flexible in terms of if you wanna do it as a slideshow, that's fine too. That you can easily do on a Chromebook, where the movement is gonna be demonstrated just by like in like short little bursts, okay? It doesn't have to be stop motion, but the idea being that I can tell what's happening by your slideshow. Not that it's jumping like really between steps really far so that it's like, well, how did they get from here to here? I can't tell what's happened here, okay? The idea here is you're gonna show me what's happened, like what is happening in the interim here? What is the process, okay? So you could do that. At the bottom, I have some ideas that I've mentioned. What did I write down here? So, for example, there's like Chrome extensions like this whiteboard extension that like just creates a virtual whiteboard, and then you can like manipulate what's on it while you're recording with Loom. So that's one way that you could do it. Or you could create some objects on screen in Google Slides and manipulate them on the screen. Or create a slideshow and literally just have steps that you go through from slide to slide. That's probably going to be the easiest for most students, I would think. Question?
What if we use the pipe cleaners that we had and record?
Totally. So any other way that you want to demonstrate it, you want to draw it on a whiteboard and record it with your phone or with a camera? Cool. You'd like to do it with pipe cleaners and show it that way? Cool. Whatever resources you have available to you, as long as you're showing the process, that's good. Okay, there's a little bit of a second thing though that you need to include, which is that you have to label what's going on screen somehow. So if you're doing with pipe cleaners, just have little cards on pieces of paper that say, you know, interface 1, so that I know what this is, okay? Like show me. It doesn't need to be anything super complex, okay? Literally just writing it on a piece of paper so that I can see it is good enough, okay? That's labeling it. That's fine. But however you want to do it, I'm really going to leave this open to you, like whatever strategy you want to use. But if you're super stuck, like you can't figure out a strategy, I can help you, okay? I can help present you with a strategy. You can use these tools if you want, but in the past, students have done this all different kinds of ways. Like I said, I've had an interpretive dance with people doing it with pool noodles and some kind of a dance, and then it's like there's like little title cards during their dance. Okay, it didn't work super well, but that someone has done that. Someone has done it in the form of song. Where also, again, you need to have a physical manipulative that I can see as labeled. Again, that's fine too. Okay, however you want to do it. Okay, you can present it kind of any way you'd like. But there are some key things that you need to include in it, okay, which is what I'm going to be looking for when I'm marking it. So, first of all, the purpose of this is that you're showing me meiosis happening in an organism, okay, a real organism. So you have to choose an organism that you're going to show me meiosis in. What is the organism? So you got to pick an organism with a 2n number. Okay, that's the full set of chromosomes is the 2n number, right? Diploid number of between 6 and 8 chromosomes. If you're looking for an organism that has between 6 and 8 chromosomes, Wikipedia's got you covered here because they got a cool article that just lists a bunch of organisms by chromosome number. I think I typed that correctly. So just look through here and find one that has between 6 and 8 chromosomes, okay? Don't pick one with an odd number. I realize that I told you that you have to have an even number for your 2n number, which makes sense because that haploid would be half, right? But there are some weird anomalies in nature where some things have odd numbers of chromosomes, and then that affects how meiosis plays out, which you don't want to go there, okay? That's too complicated and you don't want to discuss that in your presentation. So don't do that. Don't pick something that has an odd number, an odd 2n number. So just an even number, please. But anything between 6 and 8. And then when you show me this process, you're going to show it to me in that organism. So when you show the chromosomes on screen, your 2n number should be 6. There should be 6 of them on screen. And then at the end of interphase 1, there should be 6 and then an identical copy of every 6. So you should see 12 chromatids, right, after you make a copy of each one. So the key here is that you are actually showing me what it looks like in that organism, whatever organism you've chosen. Now, I said between 6 and 8. If you want to make it easier on yourself, do 6, okay? Pick an organism with 6. You don't need to pick 8 if you don't want to. I wanted to give people a little bit of flexibility because if I say 6, there's only like 2 or 3 things that have 6 chromosomes. I wanted to open it up so you could kind of choose whatever you wanted, but just as a heads up here, you don't have to pick something with 8 chromosomes. There's no like bonus marks or anything. 6 is good. Like I said, you're going to design it around the organism you chose. So the organism you chose will do meiosis somewhere on its body. Is this a male or a female of that species? You choose, okay? You don't have to show both, just pick one. But you do have to say what it is and where it is occurring, okay? So does this organism have ovaries? Then make sure you state that where this is occurring, in the ovaries of the blank, okay? Or whatever, it may have some other structure where it occurs. It's obviously very flexible in the animal kingdom, so whatever it happens to be. Okay, but you just make sure you let me know that. The whole point is this is about a specific organism that you're telling me about this process. Okay? So that's the first part here is that you want to narrow it down to a specific organism. You got to name every stage in meiosis 1 and 2. Don't forget that they have numbers that go along with them. Interphase 1, prophase 1, metaphase 1, and then interphase 2, prophase 2, metaphase 2. Make sure you label them with 1s and 2s. Okay? They all have numbers associated with them. There is no metaphase in meiosis, that's mitosis. Make sure that you show each phase occurring. Okay, so there has to be a visualization and an explanation for what's happening in each phase. The explanation doesn't have to be textual, so you don't have to show it in text, otherwise that'd be really restrictive. You can just say it, you can describe what's happening. Do me a favor, please listen to your video after you record it, to see if you can actually hear what you have said, okay? The last time I did this project, a number of people were like extremely muffled when they recorded their voice, and I had an insanely difficult time actually figuring out what people had said in their presentation. So double check that there isn't just like a scarf sitting over top of your mic or whatever happened to those people, okay? Because I need to hear what you're saying during these steps to like figure out whether you understand the process or not. So just double check. Please listen and actually watch your own video. Anyway, so you're gonna visualize and explain each step. It should be obvious from watching the difference between meiosis 1 and meiosis 2. There's obvious differences, right? Prophase 1 is completely different than prophase 2. Make sure I can see that. Okay, it should be obvious. Especially when you're doing this, you need to make sure that you cover crossing over and independent assortment. Okay? Those are the two spots during this process that create diversity in a species. It's the reason sexual reproduction is so popular in organisms in general. Okay? So you need to make sure that you are clearly showing what those processes are when they're happening, and you should talk about why they're important while they're happening. What is the purpose of this? Why is independent assortment happening? Why is crossing over happening? What does it do? Okay, we talked about that a whole bunch. It's in the note a bunch of times. Okay, if you're still unclear, let's chat. And then, oh yeah, I mentioned this already. Location of your cell should be mentioned. Plus, remember that if you're talking about a male, so if you've chosen a male, you should state somewhere that at the end, you get 4 sperm. Okay, that's consistent across the entire animal kingdom. Okay, you're gonna get 4 sperm from meiosis. Or if it's a female, you're gonna get 1 ootid and 3 polar bodies that get reabsorbed. Mention that as well. Okay, if you choose a female. Again, you can pick whatever you want to live to. All right, so that's the key stuff. I have a checklist when I go through and mark it. It's like, was this there? Yes. Was this there? Yes. Was this there? Yes. That's what I do, okay? So I'm going through the checklist to make sure you've included those things. If you've done those things, you're good, okay? So, really quickly, let me pop over to this rubric right here. So there are like 3 main areas I'm evaluating here. One is, are you naming and describing all of the phases correctly? Does this look like the phase? Have you named and described all of them? Okay, are they different in meiosis 1 and meiosis 2? And then the second part here is a real focus on crossing over and independent assortment. Have you clearly shown those in your organism? Have you shown the exchange of pieces in crossing over? Color coding works really well for that, by the way, to show little pieces being exchanged. Keep in mind that you don't have to show an exchange of DNA happening between every single homologous pair, because in real life it's random whether it happens or not. So if you only show it in chromosome 1 and 2, and you don't show it in like the others, that's fine, okay? But you do need to show it you need to show it at least happening somewhere a little bit, okay? But again, using colors is really good for that. So if you've explained those processes and why they're important and shown them clearly, that's the second part. And then the third part here is the general communication of your ideas, okay? Did you spell all of your title cards correctly? Can I hear you and understand your description for each of the phases? That's the communication part, okay? So those are sort of the 3 things. The communication is weighted a little bit less than the other 2 things. I really care about the content here. Like, do you understand the stages of meiosis and can you describe them to me? That's what it is. And, you know, are you communicating them well to another person? Again, the idea here generally is that you're showing this process to, let's say, somebody who's going to take this process next semester. You are explaining the process of meiosis to them. Okay, that's sort of the level that you're looking at, okay? So think about that at the end. Like, did you actually explain to them what this process is and what's happening? Or if you watched this 3 weeks ago, would you be like, I have no idea what just happened? If that's the case, that's not good, okay? That's not where we're headed for here. Okay, so that's the basics of this. When you're done, you're just going to attach it to your assignment and submit. In this case, it could be submitted in a lot of different ways, right? Like it could be a Loom video that you record, then you just give me the link there and submit it. It could be a video file. Maybe you've uploaded a private video to YouTube and you're gonna give me a link to your YouTube video, whatever, okay? The one thing I will ask is before you submit, please test your link. That was another thing that we had a huge problem with last time we did this, which was that people submitted something and like, I don't know, at least 5 people's links didn't work, okay? Check, you know, does it work, the thing that you sent me? You know, just to make sure you actually get something out of this. Again, ideally, you would be done this by the end of the day tomorrow. There's a lot here. This is going to take a little while for you to put together. You pretty much have the entire day to work on this, okay? Minus the quiz, and there's like that short reading activity that will probably take you 25 minutes in the morning, okay? So the rest of the day is yours to work on this. Realistically, again, I'm not going to get to marking this until the weekend. So if you're heading in on Friday, that's fine. I can't make the due date Friday because we're not allowed to make due dates outside of like our days. So like that's your time in your other course, right? So technically this is due tomorrow at midnight. But if you need until Friday, that's fine. I can't put the deadline there because that's during your other course time. So ideally it shouldn't take you that long, but if it does, that's fine. Not a huge deal. Any questions about this assignment from people at home too? Feel free to ask. I think it's fairly straightforward. Okay. Again, it's wide open how you want to show me this. Like you can use any kind of tool you want pretty much. Use a camera or whatever. Okay? Okay, so then I will, I'll stop talking now. And getting back here to what you're working on. Oh, right, I'm gonna take the homework out. So let's do that quickly. I'll try not to take up too much of your time. I realize that people want to work on this, so I'll go quick. Oh, that's not what I want to do. What did I just do here? This thing? There we go. Okay. So first of all, in general, regarding blood types, are there any questions in terms of how this works? How you get various blood types and how you figure out who can donate to who and who can receive blood from whom. You guys are okay with that in general? Okay. I feel like in the video I did an okay job with that, but yes, question.
We don't talk about positives and negatives, do we?
No. So with the rhodopsin factor, the positive and negative part, you can basically just ignore for the purpose of this course. It's an additional level of this. So it's this plus another gene that also participates, but that's too complicated and we don't care about it for this course. So, but it does exist.
Yeah.
Okay, I'm gonna run through these right now. So, ooh, Brad Pitt. This is a Miss Dick question. I borrowed this from her. Pretend that Brad Pitt is homozygous for the type B allele, and Angela Jolie is type O. Oh, are they together anymore? No, definitely not. Whoops. That's awkward. Oh, that probably made Miss Dick very sad. What are all the possible blood types of their baby? Okay, so again, when you're starting one of these problems, you need to use let statements. Okay? You always start a genetics problem like this with let statements and describe what strategy you're going to use to name all of these different alleles, okay? In this case, we're talking about, wait, no, they're codominant, right? Codominant. Tell me, all of this is incorrect? Oh, that makes me so mad. I bet you I took this straight out of the text, that's why. Whoops. Whatever, I'm gonna leave this. I'm gonna leave this notation because it's exactly the same as the text and it's confusing otherwise. But they use capital letters here for A and B, but technically this is codominant, so they should be small a and B. But I think this is actually like a.
You said that in the video. You said that for the purpose of whatever you're gonna use.
Okay, fair enough. Fair enough. It's like that in the text, and this also might be like industry standard for blood typing. So what? I'm gonna leave all this the same. So anyway, A and B. Those are codominant, so I've used the correct notation here for codominance. And then I didn't take this up in the video, did I? I don't think so. These questions?
Yeah, you did.
These ones as well? I thought I just did these ones. I did these too. Oh, I forgot that I did that. Okay, cool. Save me some time. All right, you've already seen all this. Fantastic. Then I'll let you guys get to work then. If anyone does anyone have any questions about these before I just say forget about it?
Should we work on the project first or just do the reading questions first?
How you want to like arrange this day is kind of up to you. You got to do the genetic illnesses thing because there are parts of that are potentially testable on the quiz. However, the assignment is really where all the marks are at. So I'll let you balance your time however you want. And not that the quiz is not worth anything. I am going to count it. It's just not weighted as heavily as the assignment. So again, you decide how you want to allocate your time. But the assignment is obviously going to take you a lot longer as well. So, okay, so I'll let you get to that. You can really do it in any order you like. Again, the reading is on page, where is it? 204 to 207. And if you're looking for the little blurb that's in your notes, it's on page 14 in your notes. This is it right here. Okay, so you're doing the reading. There's a couple questions from the text, and then you have the rest of the day to work on your Meiosis Live assignment. If you need anything, people at home, I don't understand something, you're looking for a little bit of help with something, let me know, please. Say something in the chat. I'll leave it open all day so that we can jump into a video conference or something and I can answer your questions properly. And please in here, if you're stuck on something or don't understand something, please say something, okay? You don't need to be saying, I'm here to help you. I'm not busy up here or anything, okay? Let me know, I'm here to help. Okay, cool, then I'll let you get going.